Hi, welcome to Kavita's NEET classes. In this lecture, we shall discuss a simple topic, the structure of DNA. So now many of you all might be wondering, what's so special about the structure of DNA? After all, we have studied it right from our school days. So we all know that it was James Watson and Francis Crick who proposed the double helical structure of DNA. And what are purines, what are pyrimidines, what is adenine, guanine? So what's so special about that? There's nothing special about the structure of DNA as such, but the approach which you have to follow for a competitive examination should be completely different from what you have been doing all these days preparing for a board examination. So be it this topic, that is the structure of DNA, or any other topic, you would have so far approached it with the point of writing a good answer in your board examination or your school exams, isn't it? You would have studied about what is the DNA, what is the structure, what are polynucleotide chains, what is the composition of the DNA. You would have learned how to make a diagram of the DNA and label what is a major groove, minor groove, and so on. But here, when you're preparing for a competitive examination like NEET, your approach should be different from that of what you've been doing or how you've been approaching your board examination. Here, you're not writing essay type answers. Say you're not writing a 5-mark answer on the structure of DNA or a 10-mark answer. Here, you have the answers in your question paper itself. So, this is an objective type question. This examination is all about thinking objectively. You are given a question followed by 4 different options, and you have to choose the best option or the right option for that particular question. And that too, you have to do it within a limited time frame. So this is what makes the examination tough, isn't it? So when asked to write about the structure of DNA, all of you definitely can write a very good answer for 5 marks or 10 marks. But when given a question asking where is DNA present other than the nucleus in a prokaryotic cell or in a eukaryotic cell, and given 3 or 4 options, and when you read all these 3 or 4 options, you will have slight confusion thinking which one is the right one. So here, whenever you are preparing for a competitive examination, nobody is going to ask you how much marks or what percentage of marks you are scoring. So whether you score distinction or whether you're scoring first class, second class, or you're just having a just pass marks, that is not going to matter here. In a competitive examination, it is just like running a race. You have to outperform the other competitors who are also taking the examination. So here in a competitive examination scenario, you'll have to think objectively. Whenever you read any paragraph or any topic or even a single line, you need to analyze, not just understanding the concepts and memorizing few facts. Yes, definitely here and there, few facts like the name of the researchers or the person who discovered, few factual things we have to remember and we have to memorize. But in addition to that, we have to have a very clear understanding of the concepts. So conceptual clarity plays a very important role, and in addition to that, you should also analyze and you must be able to guess or make questions out of the topics whatever you are reading, isn't it? Say you are reading a paragraph, you must think like an examiner and try to frame questions out of that. You must try to frame questions thinking that you are an examiner and try to understand, okay, what kind of questions can an examiner make from this particular topic. So this will help you in approaching this exam objectively and score much better and achieve your goals, isn't it? So in future, we will be discussing more about the tips, tricks, how to approach various questions and so on. As far as this lecture, let us begin with the structure of DNA. So even before we start discussing about the structure of DNA, the components of the DNA, let's just go through the basics of where is the DNA present in the prokaryotic cell, eukaryotic cell, where it occurs, and so on. And then we shall start discussing about the structure of DNA. The occurrence of DNA. Where does the DNA occur? So even before that, DNA. What is DNA? DNA is deoxyribonucleic acid, isn't it? DNA is deoxyribonucleic acid. So what is DNA? DNA is the genetic material which is responsible for transmission of genetic information across generations. DNA is the genetic material in most of the organisms. However, we also have certain organisms which have RNA as their genetic material. Say for example, the retroviruses. We have viruses which have RNA as the genetic material and they are called as retroviruses. So most of the organisms have DNA as the genetic material. The genes are present inside the DNA and it is the genes which are responsible for our characters or traits. Characters or traits can be very simple, like the shape of your eyes, the color of your eyes, the shape of your nose, ears, your skin color, hair texture. Any of these can be characters or traits. So your skin color, your eye color or the shape of your nose is determined by the genes which you inherited from your parents, isn't it? So whatever genes you have inherited from your father or mother determines your eye color or your eye shape or your nose shape or your skin color and so on. So it is the genes which are responsible for transmission of the information from one generation to the other generation. And the genes are contained in this genetic material called as DNA. Okay, that is deoxyribonucleic acid. The genetic material in most of the organisms is DNA. However, we do have certain organisms which have RNA as the genetic material. So what is RNA? RNA is the ribose nucleic acid. RNA is ribose nucleic acid. Where does the DNA occur in a cell? So we know we have two kinds of cell, the prokaryotic cell and the eukaryotic cell. The prokaryotic cell is the primitive cell which is present in lower organisms like certain bacteria, amoeba, euglena and so on. So prokaryotic cells are mostly found in primitive organisms, whereas the eukaryotic cells are the ones which are more advanced and it has well-defined organelles. So we shall be discussing more about the difference between the prokaryotic cell, eukaryotic cell, etc. in our future lectures. First of all, in a prokaryotic cell, the DNA is present as circular structures freely suspended in the cytoplasm. Here you can see this, isn't it? These coiled structures, circular coiled structures which occur freely in the cytoplasm in a prokaryotic cell. This is called as nucleoid. This is called as nucleoid or nucleoid DNA. In addition to this nucleoid DNA, we have circular DNA molecules which are capable of self-replication. These are called as the plasmid. Okay, so this is the nuclear DNA which is freely suspended in the cytoplasm and this is called as nucleoid DNA, and in addition to this we also have extranuclear DNA. Extranuclear DNA appearing as circular DNA molecules in the bacterial cell. These circular DNA molecules are capable of self-replication. That is, they can replicate on their own and they have been used as vector molecules in recombinant DNA technology or genetic engineering experiments. So how exactly this plasmid molecules are utilized in genetic engineering experiments? We shall discuss that in our future lectures. So, in addition to the nucleoid DNA, in a bacterial DNA, in a bacterial cell, you have circular DNA molecules which are capable of self-replication called as plasmids. Got it? Next, moving on to the eukaryote. The eukaryotic cells are present in higher organisms, isn't it? So all plants, animals and higher organisms have eukaryotic cells which have very well defined organelles. So how is the DNA present in a eukaryotic cell? In a eukaryotic cell, you can find that the DNA is present as a complex along with proteins called as histones. So you have a histone octamer. You have 8 histone proteins which together form a histone octamer around which the DNA molecule is circled like this. So we get a chain of DNA with beads of histomers or beads of histone proteins. It appears just like this. You get histone proteins and it looks like a beaded chain. So this is how it is present in eukaryotic cells. What happens is this DNA along with the histone proteins is compacted in the form of chromosomes and chromosomes are present inside the nucleus. Got it? So DNA along with the histone proteins are compacted to form chromosomes which are present inside the nucleus. So nucleus is a very well defined organelle which has a nuclear membrane consisting of nuclear pores, and in a eukaryotic cell all the other organelles also are very well defined. So it is an advanced form, isn't it? So in eukaryotic cell, in addition to the chromosomal DNA, do we have DNA in any other organelles? Yes. In addition to chromosomal DNA, in eukaryotic cells you can find DNA in mitochondria. So mitochondria are also called as the powerhouses of the cell, isn't it? So this is the mitochondria. So in mitochondria also you can find extra chromosomal DNA. In addition to this, we can also find extra chromosomal DNA in plastids. Okay. So now that we discussed about the occurrence of DNA in a prokaryotic cell and an eukaryotic cell, now we must try to guess what kind of question can my examiner make from this whatever I have studied. So say your examiner can come up with a question like this. So the question can be like this. What are the additional DNA other than the nucleoid DNA that is present in a prokaryotic cell? Okay. So what is the other additional, apart from nucleoid DNA, what are the other additional DNA that is present in a prokaryotic cell? So the question can be, what is the additional DNA that is present in a prokaryotic cell apart from this nucleoid DNA? And the answer options can be, first one can be plasmid, and the second option can be mitochondrial DNA, and the third option can be plastids, okay, and the fourth option can be all of the above. So now what will be the right answer? The question asks, what is the extra DNA that is present in a prokaryotic cell? So the extra DNA apart from this that is present in a prokaryotic cell is the plasmid DNA. So the right answer will be plasmid. Whereas suppose if the same question can be asked with respect to an eukaryotic cell. So if they ask what are the DNA apart from the chromosomal DNA that is present in a eukaryotic cell. And you have the same 4 options. So now what is the right answer? So instead of prokaryotic cell, if they are asking about, extra chromosomal DNA that is present in a eukaryotic cell. So what will be the answer? It cannot be plasmid because plasmids are present only in prokaryotic cell. So here the right answer can be, say the 4th option is both 2 and 3. Both 2 and 3. That is, here we know that in a eukaryotic cell, extra chromosomal DNA is present in mitochondria. So 2 is the right answer. And it is also present in plastids. Right? So second one is also right and third one is also right. However, you can have only one right answer. So the 4th option, which includes both of them, will be the right option over here. Okay, so now are you able to understand how we have to think and formulate questions when we are studying any particular topic? This will help you a lot when you are facing the examination. So we shall further move on. Now that we finished discussion about the occurrence of DNA in a prokaryotic cell and an eukaryotic cell, now we shall discuss about the composition of DNA. What is the DNA composed of? DNA is composed of 3 components. Okay, DNA is composed of 3 components. First one is the pentose sugar. Second one is the phosphate group. And the third one is nitrogenous base. DNA is composed of 3 components. First one is the pentose sugar, second one is the phosphate group, and third one is the nitrogenous base. This is the phosphate group, isn't it? This is the phosphate group. And this is the deoxyribose sugar. So the sugar in DNA molecule is deoxyribose, whereas the sugar in a RNA molecule is ribose, isn't it? So RNA is called as ribonucleic acid and DNA is called as deoxyribonucleic acid, isn't it? So what is the difference between ribose and deoxyribose, isn't it? This can also appear as a question. So what is the difference between ribose, which is present in RNA, and deoxyribose, which is present in DNA? So here you can see that in the second carbon atom we have H, that is a single hydrogen molecule. That is why it is called as deoxyribose, whereas in a ribose sugar which is present in RNA, at the second carbon atom, instead of this we will have a hydroxyl group, that is OH. That is the difference between deoxyribose and ribose. Okay, at the second carbon atom if you have H, then it is deoxyribose, whereas if you have a hydroxyl group, that is OH, in this position, it becomes a ribose sugar. So the DNA is composed of pentose sugar, phosphate group, and nitrogenous base. So what are the nitrogenous bases? We have 2 kinds of nitrogenous base. One is the purine and the other is pyrimidine. Purine are double ring structures. Under purines, we have two types. One is adenine and the other one is guanine. So we have two purines. One is adenine, which is represented by the capital letter A, and guanine, which is represented by the capital letter G. And under pyrimidines, we have cytosine and thymidine. Cytosine and thymine. Sorry, it's not thymidine, it is thymine. So, under pyrimidines, we have cytosine and thymine. Pyrimidines are single ring structures, whereas purines are double ring structures. Okay, keep this also in mind. This can also appear as an option somewhere. So, most of the time what happens is, you might not exactly know the right answer for a question, but based on your understanding of various concepts, you will at least be capable of eliminating the wrong options. So when you eliminate the wrong options, obviously you will land at the right option. So understanding each of these will definitely help you tackle such kind of questions. So purines are double ring structures. So purine are adenine and guanine, and we have pyrimidines, which are single ring structures. That is, we have two pyrimidines, that is cytosine and thymine. So these are the four nitrogenous bases which are present in DNA, that is adenine, guanine, cytosine and thymine. In RNA, we have four nitrogenous bases: adenine, guanine, cytosine and uracil. Instead of thymine, so in DNA we have thymine, in RNA in place of thymine we have uracil. Okay. So did you understand so much? That is the components of DNA. We have three components of DNA. First is the pentose sugar, then the phosphate group and the nitrogenous base. There's no particular order for this. Okay. Next. So how are these three components of DNA, that is phosphate group, ribose sugar and nitrogenous base, linked with each other and arranged in the DNA? First, this is the deoxyribose sugar. The deoxyribose sugar is attached to the nitrogen base. This is the nitrogen base and this is the pentose sugar. Okay. The nitrogenous base and the pentose sugar are attached together by a glycosidic linkage. Shortly, I'll be telling you what a glycosidic linkage means. So, a sugar molecule is attached, a nitrogenous base is attached to the carbon 1 atom of the sugar molecule through a glycosidic linkage, and this combination of the nitrogenous base with the ribose sugar is called as a nucleoside. Okay. So I'll write it down here. Nitrogenous ribose sugar plus nitrogenous base. The ribose sugar plus nitrogenous base is called as a nucleoside. Okay. So ribose sugar plus nitrogenous base is a nucleoside, and the nucleoside plus the phosphate group, okay, that is called as the nucleotide. Sorry, it's not phosphate, it's phosphorus. Okay, so keep this in mind always. The ribose sugar plus nitrogenous base gives a nucleoside. And nucleoside plus the phosphorus group gives the nucleotide. Okay. So here you can see very clearly, this is the nitrogenous base, this is the nitrogenous base and the pentose sugar. These two together form the nucleoside. Pentose sugar plus nitrogenous base form the nucleoside. To that, if you add the phosphate group, sorry, it is phosphate group. So to the nitrogenous base and the pentose sugar, which is called as a nucleoside, if you add the phosphate group, it becomes the nucleotide. Okay, ribose sugar plus nitrogenous base gives nucleoside. Nucleoside plus phosphate groups gives nucleotide. So did you understand what's the difference between a nucleoside and nucleotide now? So you can have questions making you confused between this also. So you might get a question asking, what are the components of a nucleoside? So the options might be, first option can be, it is the pentose sugar plus nitrogenous base. Okay, next option can be pentose sugar plus phosphate group, and the third option can be pentose sugar plus nitrogenous base plus phosphate group, and the fourth option can be all of the above. Okay, say the question is, what is a nucleoside or what are the components of a nucleoside? So in this case, if these are the four options, which one would you choose? So now you might get a confusion whether it is pentose sugar plus phosphate or pentose sugar plus nitrogenous base or all the three. So all these three, that is pentose sugar plus nitrogenous base plus phosphate group, is a nucleotide. It is a nucleotide. It is not a nucleoside. Okay, so if the question is which one is a nucleotide, then this will be the answer. Whereas if the question is what are the components of a nucleoside, then your answer should be pentose sugar plus nitrogenous base. Got it? So as in how you keep reading, you need to keep guessing questions like this and make yourself much more confident when you get the questions so that you can answer it. Fine. So now you know what is the difference between a nucleoside and a nucleotide. DNA or deoxyribonucleic acid is a polymer of nucleotides. It is a polynucleotide chain. So you can find many nucleotides attached to one another to form a chain. So it is called as a polymer of nucleotides. In DNA, we have two important bonds or linkages. One is the glycosidic bond and the second one is the phosphodiester bond. What is a glycosidic bond? Glycosidic bonds are linkages between the nitrogenous base and the pentose sugar. So here you can find that this is the pentose sugar, isn't it? This is the pentose sugar. It is linked to a nitrogenous base. So here this linking between the nitrogenous base and the carbon 1 atom of the pentose sugar is called as a glycosidic bond or a glycosidic linkage. Both glycosidic bonds and glycosidic linkages are formed due to condensation reaction, which is involved with elimination of water. Okay, so in DNA we have two kinds of bonds, that is glycosidic bond and phosphodiester bond. What is a glycosidic bond? Glycosidic bond is a linkage or a bond between the pentose sugar and the nitrogenous base. So the glycosidic bond, that is the link or the bond between the pentose sugar and the nitrogenous base, gives rise to a nucleoside, isn't it? Pentose sugar and nitrogenous base give rise to a nucleoside. So when the nitrogenous base is a adenine, okay, when the nitrogenous base is adenine, so here you can see we have the deoxyribose sugar molecule which is attached with adenine nitrogenous base. So then what is the nucleoside which we get? The nucleoside is deoxyadenosine. Okay, so what is deoxyadenosine? Deoxyribose sugar plus adenine, which is linked together by glycosidic linkage. Did you understand? So now you must not get confused. So under nucleosides we have 4 types, that is deoxyadenosine, deoxyguanosine, deoxycytidine and deoxythymidine. So what is deoxyadenosine? Ribose sugar linked with adenine nitrogen base by the glycosidic bond, that gives deoxyadenosine. Similarly, the deoxyribose sugar linked with guanine, the nitrogenous base guanine linked with the pentose sugar deoxyribose, gives rise to another nucleoside called as deoxyguanosine. So the ribose sugar is the same in all of them. The only difference is in the nitrogenous base. So one ribose sugar is linked with one nitrogenous base. When the nitrogenous base is adenine, it gives rise to deoxyadenosine. When the nitrogenous base is guanine, it gives rise to deoxyguanosine. And then when the nitrogenous base is cytosine, it gives rise to deoxycytidine. And when the nitrogenous base is thymine, it gives rise to deoxythymidine. And all of these 4 are nucleosides. Got it? We have 4 kinds of nucleosides in DNA, that is deoxyadenosine, deoxyguanosine, deoxycytidine and deoxythymidine. So what happens in the case of RNA? Okay, in RNA it is adenosine, where in RNA what happens is, instead of deoxyribose sugar, here we have a ribose sugar which gives rise to adenosine, guanosine, cytidine, and in the place of thymine we have uridine. Can you recall? In RNA we do not have thymine, we have uridine, isn't it? So what are the nucleosides in DNA? Just let's list it out. So the nucleosides in DNA are deoxyadenosine, deoxyguanosine, deoxycytidine and deoxythymidine. What are the nucleosides in RNA? In RNA we have adenosine, we have guanosine, we have cytidine, and the last one, instead of thymine we have uracil, isn't it? So there we have uridine. So now you might get a question, what are the nucleosides present in DNA or what are the nucleosides present in RNA? So suppose you get these options and in the 4th one, instead of uridine, if it is something else. So now you must be able to have a clear picture as to what are the nucleosides in DNA and what are the nucleosides in RNA, so that you can arrive at the right answer. So did you understand about the glycosidic linkage? Glycosidic linkage is a linkage between the pentose sugar and the nitrogenous base. The second bond or the linkage that is present in DNA is the phosphodiester bond. What is the phosphodiester bond? Phosphodiester bond is the one which connects adjacent nucleotides. Okay, so when we have adjacent nucleotides A, T, C, G, A, T, T, so it is the phosphodiester bonds which connect them together to form a polymer of nucleotides, or it is the phosphodiester bond which forms the nucleotide chain. See, you can see here, this is the pentose sugar, this is the phosphodiester bond. So this is one ester bond and this is the other ester bond, and this complete bond is called as the phosphodiester bond. So here we have one pentose sugar connected to one nitrogen base. So this is one nucleotide, okay, and this is the second nucleotide. So the nucleotides, one nucleotide is connected to the next nucleotide through phosphorus by a phosphodiester bond. So why do we call it as a phosphodiester bond? Because each phosphate or hydroxyl bond is an ester bond, and the linkage between the two is called 3' phosphodiester linkage or bond. So here you can see this is one ester bond, right? This is one ester bond and this is another ester bond, and here we have phosphorus molecule, isn't it? So since each phosphate hydroxyl bond is an ester bond, the linkage between these two is called as 3' to 5' phosphodiester linkage or bond. So now did you understand what is a glycosidic bond and what is a phosphodiester bond? Glycosidic bond is the bond between a pentose sugar and a nitrogenous base, whereas phosphodiester bond is the bond which connects two adjacent nucleotides, and both of them are formed by the condensation reaction which involves elimination of water. So these are the two important bonds in the DNA structure. So here I can show you. See, this is the phosphodiester bond, isn't it? This is one nucleotide. Okay, this is the nucleotide adenine, isn't it? This is adenine nucleotide and this is thymine nucleotide. Okay, and they both are connected by this phosphodiester bond. So as I already told you, phosphodiester bond is the one which connects adjacent nucleotides with a phosphate group. This is the phosphodiester bond. And what about the glycosidic bond? Here you can see this is the pentose sugar, and the pentose sugar is connected to the nitrogenous base adenine over here. So this is the glycosidic bond. This is the glycosidic bond and this is the phosphodiester bond. And in addition to this, we also have hydrogen bonds. That is hydrogen bonds between adenine and thymine and guanine and cytosine. Okay, that is the nucleotides are bonding with the opposite base pairs. So this is called as one base pair, which we shall be discussing shortly. Okay, so this is the base pair. So in a DNA, always adenine base pairs with thymine with two hydrogen bonds. You can see the two hydrogen bonds here. And then guanine always base pairs with cytosine with 3 hydrogen bonds. Got it? So we shall be discussing this further, do not worry. So the 3 bonds are glycosidic bond, phosphodiester bond and the hydrogen bonds. This is how a double-stranded DNA molecule looks like. Okay, this is the double-stranded polynucleotide chain. Here you can find one strand of DNA moving in 5' to 3' direction and the other strand of DNA in 3' to 5' direction. So now we all must be wondering, what is the length of the DNA? How long is it? So how do we measure the length of the DNA? So DNA is a long polymer of deoxyribonucleotides, isn't it? Its length is defined as the number of nucleotides or a pair of nucleotide referred to as base pair. So how do we measure the length of DNA? We measure it in the form of base pairs. So here you can see this is one base pair. AT is one base pair. And then this one is the second base pair, another AT. So this is one base pair. This is another base pair. So like this we can count 1, 2, 3, 4. That count gives us the length of DNA. Got it? How do we measure DNA? How do we know what is the length of DNA? So we measure the nucleotides or a pair of nucleotides. So this is one pair of nucleotides, that is AT. This is another pair of nucleotide GC. So here in this DNA you can find there are 4 nucleotide pairs, isn't it? This is 1, this is 2, this is 3 and this is 4. So there are 4 base pairs, 4 nucleotide base pairs. So the length of this DNA is 4. So like this we can find that the length of DNA varies from organism to organism. Say for example in E. coli or Escherichia coli, the length of the DNA is 4.6. In E. coli or Escherichia coli, the length of the DNA is 4.6 into 10 to the power of 6 base pairs, and base pairs is represented by small letters B and P. Okay, so like this the length of the DNA varies from species and organism to organism. What is Escherichia coli? It is a simple bacteria which is present in our own colon, isn't it? It is present in all of us. Fine. So the length of the DNA or the length of the DNA in E. coli bacteria is 4.6 into 10 to the power of 6 base pairs. And what do I mean by polarity of DNA? Polarity means you can see that we have 2 strands of DNA which are coiled or twisted with each other, and in that you can find that one strand of DNA runs in the 5' to 3' direction and the other one runs in the 3' to 5' direction. So in the 5' end you can find that we have a free phosphate group. See here, see this strand, okay, this strand of DNA is in 5' to 3' direction and this strand of DNA is in 3' to 5' direction. So when we say polarity, 5' end means it has a free phosphate group at this position, and at the 3' position you have a free hydroxyl group. This is what we mean by polarity in the DNA. So 5' end means there is a free phosphate group and 3' end means there is a free hydroxyl group. Got it? Now finally coming to the structure of DNA. In 1950 to 1953, several researchers did research and they gave us significant knowledge about the DNA. So many researchers, scientists like Edwin Chargaff, Maurice Wilkins, Rosalind Franklin, James Watson and Francis Crick, based on their studies, gave us a lot of knowledge about the structure of DNA. However, in 1953, James Watson and Francis Crick proposed the double helical structure of DNA. In 1953, James Watson and Francis Crick gave us the double helical structure of DNA based on 2 major investigations. First one was the Chargaff rules and second one is the X-ray diffraction pattern of DNA. What are Chargaff rules? The Chargaff rules has three important rules. First one is the purines and pyrimidines are always in equal amounts. The purines and pyrimidines. What are the purines? Adenine and guanine, isn't it? And what are the pyrimidines? Cytosine and thymine. So the amount of adenine and guanine will always be equal to the amount of cytosine and thymine. This is the first rule of Chargaff rule. That is, adenine, the amount of adenine and guanine will always be equal to that of the amount of cytosine plus thymine. Okay, this is the first rule. Now moving on to the second rule. The amount of adenine is always equal to that of thymine and the amount of guanine is always equal to that of cytosine. That is, the amount of adenine is always equal to that of thymine and the amount of guanine is always equal to that of cytosine. So this gives us the second Chargaff rule, that is, adenine and thymine amounts are always equal and guanine and cytosine amounts are always equal. Now moving on to the third rule of Chargaff's rule. It states that the base ratio of adenine and thymine, that is A plus T, is always constant for a species. The base ratio of adenine and thymine is constant for a species. However, it may vary from one species to another. Say for example, we all belong to Homo sapiens, isn't it? So the base ratio of A plus T will be same in all Homo sapiens. However, the A plus T will be different in a monkey, or it will be different in a dog or cat which belong to another species. However, among the members, all the members belonging to a particular species, the base ratio of A plus T will be constant. And this helps us to identify which species that particular animal belongs to. Got it? So that the third Chargaff rule says that the adenine plus thymine base ratio will be constant for a species. Okay, so if among all the members belong to a particular species, the adenine plus thymine ratio will be constant. Did you understand the three Chargaff rules? So it is based on the Chargaff rules and the X-ray studies that James Watson and Francis Crick were finally able to propose the double helical structure. That is why Chargaff's rule is very important. So you might get this also as a question. So what are the three rules of Chargaff's rules? And they might give you these equations. So then there might be some wrong equation also. Say for example, in the last, apart from all these three options, you might get one more option saying the base ratio of guanine and cytosine is always constant for a species. Now say the question is, which of the following is not a Chargaff rule? Okay, so the first option will be A plus G equal to T plus C. So this can be the first option. This can be the second option. And A plus T is constant for a species. This can be the third option. And the fourth option can be G plus C is constant for a species. So now if the question is, which is not a Chargaff rule, which is not a point in the Chargaff rule, then this will be the right answer, isn't it? Because all the above three are points in the Chargaff rule. All the three are important rules of the Chargaff rules. Got it? So this is how you have to find out questions and answer it so that you do not get confused in the examination. In addition to the Chargaff rule, the X-ray diffraction pattern of DNA given by Maurice Wilkins and Rosalind Franklin in 1953 also was helpful for Watson and Crick to propose their double helical structure. So in 1953, Maurice Wilkins and Rosalind Franklin did X-ray diffraction studies and they got the pictures of crystalline DNA. Based on the pictures which they got about the DNA, they concluded that DNA is a long molecule consisting of two similar DNA strands running in parallel and helical manner, where successive nucleotides occur at intervals of 0.34 nanometer. So now you might even get a question, is it that only Watson and Crick said that the double helical structure, is it only them who observed the double helical structure, or whether anybody else prior to them who said that DNA has two polynucleotide chains which are running in anti-parallel direction? So then it is not just Watson and Crick, even Morris Wilkins and Rosalind Franklin, based on the pictures of the crystalline DNA. So this is the picture which they saw. Okay, so based on the pictures of the crystalline DNA, they also concluded that DNA is made up of two strands which are parallel to each other. So that also can be an option, isn't it? So they said that based on the pictures of the crystalline DNA, they concluded that DNA is a long molecule consisting of two similar DNA strands running in parallel direction, and they have nucleotides occurring at successive gaps, and those intervals are of the measurement 0.34 nanometer, or we can also call it as 34 angstrom unit. Isn't it? 0.34 nanometer is also 34 angstrom unit. So based on the X-ray diffraction pattern of DNA, and also based on the these two important investigations, that is the X-ray diffraction pattern of DNA and the Chargaff rules, finally in 1953, James Watson and Francis Crick proposed the double helical structure of DNA. So what are the salient features of the double helical structure of DNA? First and foremost, DNA is made up of two polynucleotide chains or strands. So these are the important features of the double helical structure of DNA proposed by James Watson and Francis Crick in 1953. So first one is, the DNA is composed of two polynucleotide chains or strands. The DNA is composed of two polynucleotide chains or strands. So here you can see DNA is made up of two chains or strands, isn't it? One strand runs from 5' to 3' direction, and the other one runs from 3' to 5' direction. Then there are glycosidic and phosphodiester linkages. There are glycosidic and phosphodiester linkages. So already we have discussed what is a glycosidic bond, what is a phosphodiester bond, and where they occur in the DNA. Then the DNA is a duplex. DNA is a duplex. So if you need to picturize how exactly the DNA will look like, just take two threads, tie a knot at one end and fix that to a surface or somewhere, and then hold the other two strands and rotate it in a right-handed fashion like this. So the resultant, whatever you get, that is exactly how a DNA looks like. Okay, DNA is rotated in a right-handed fashion. So in a DNA duplex, you can find that the one twist or one turn is equal to 34 angstroms or 3.4 nanometer. Isn't it? So this is one turn of the DNA. From here up to here, this is one turn of the DNA, and in that one turn, you can find approximately 10 base pairs. So in every turn of the DNA, there are approximately 10 base pairs, and this one turn measures 34 angstrom unit or 3.4 nanometer. Okay, and then we have two kinds of grooves in DNA. This is the major groove and this is the minor groove. Can you see this? Here again you can see this is the major groove. This region over here, this is the major groove, and this region over here, this is the minor groove. And then the diameter of DNA, if you see the thickness of the DNA, the diameter, that is the distance from here to here, that is 20 angstrom units or 2 nanometer. Okay, so this region is called as the diameter. These are very important. You definitely need to remember this. So the diameter of DNA is 2 nanometer or 20 angstrom units. And then this is the major groove, this is the minor groove. Just based on the structure, okay, these numbers itself, you can get different kinds of questions. So keep this in mind so that you remember this. The diameter of DNA is 2 nanometer or 2 nanometer or 20 angstrom unit. And then in per turn, okay, per turn of the helix, you have approximately 10 base pairs which measures, and this measurement is 34 angstrom unit or 3 nanometer. Okay, and then the diameter is 20 angstrom unit or 2 nanometer. So there are two strands which are running in opposite direction. Okay, one is in 5 prime to 3 prime direction and the other one is 3 prime to 5 prime direction. And the coiling is plectonemic. Coiling is plectonemic. What do we mean by plectonemic? That is, the two strands cannot be separated without completely unwinding them. The DNA is plectonomic. Remember this word. Please do not get confused with this word when you get it in the option. What do we mean by plectonomic? Plectonomic means the two strands of DNA cannot be completely separated without unwinding. So here you can see there are two strands. Until and unless you unwind them completely, you cannot separate the two strands, isn't it? That is what we mean by plectonomic. Okay, so DNA is plectonomic. And then the backbone of DNA. What is the backbone of DNA? The ribose sugar. Okay, the ribose sugar and the phosphate group forms the backbone of DNA, and from that backbone you can find the nitrogenous bases projecting inwards. I will show you here. See, here you will have the ribose sugar and the phosphate group. Okay, so here completely, here you can find the ribose sugar and the phosphate groups, whereas here you can find the nitrogenous bases projecting inside. So it is the ribose sugar and the phosphate group which acts as the backbone of DNA, and from this backbone you can find the nitrogenous bases projecting inwards. So in this picture, this is T, this is thymine, this is adenine, and this is guanine, and this is cytosine. So orange is guanine, yellow is cytosine, green is thymine, and blue is adenine. So here this is the backbone of DNA. So in this backbone you will have pentose sugar and phosphate group, and then from this backbone you can find the nitrogenous bases projecting inwards. Got it? Did you understand? Backbone of DNA. What is the backbone of DNA and how the nitrogenous bases project inwards. Then polarity. I already told you about the polarity of DNA, isn't it? So here in DNA we have two strands, one strand running from 5 prime to 3 prime direction and the other one in the opposite, that is 3 prime to 5 prime direction. At the 5 prime end we have a free phosphate group, and at the 3 prime end we have a free hydroxyl group. This is what we mean by polarity of the DNA. And then here in the double helical structure of DNA, Watson and Crick also spoke about the complementary base pairing. So you can find that in DNA, though we have purines and pyrimidines, isn't it? Adenine and guanine are purines, cytosine and thymine are pyrimidines. You can find that one purine combines with another pyrimidine. So adenine always base pairs with thymine with 2 hydrogen bonds, and then guanine always base pairs with cytosine with 3 hydrogen bond, and this is what we call it as complementary base pairing. Did you understand what we mean by complementary base pairing? Adenine always base pairs with thymine. You can see this in the picture over here. So this is adenine. This is adenine. It is base pairing with thymine. This is adenine. It is base pairing with thymine. And then here you can see guanine is base pairing with cytosine. Here again guanine is base pairing with cytosine. So adenine always base pair with thymine with 2 hydrogen bonds, and guanine always base pairs with cytosine through 3 hydrogen bonds. This is what we call it as complementary base pairing. And then antiparallel strands. So what do we mean by antiparallel? That is, we have 2 strands, that in the one in 5' to 3' direction, and the other one is parallel to that, but it runs in the opposite direction, that is 3' to 5' direction. So now shall we summarize the double helical structure of DNA once again? So according to the double helical structure proposed by James Watson and Francis Crick in 1953, DNA is composed of 2 polynucleotide chains. The 2 chains have antiparallel polarity, that is, one chain runs from 5' to 3' direction and the other chain runs from 3' to 5' direction. And the backbone of each polynucleotide chain is composed of ribose sugar and the phosphate group, and from the backbone you can find the nitrogenous bases projecting here. And then a purine always base pairs with the pyrimidine. Specifically, adenine always base pairs with a pyrimidine called as thymine, and guanine, which is a purine, always base pairs with the pyrimidine called as cytosine. This is what we call it as complementary base pairing. The double helical chain, the double chain is coiled in a helical fashion. Okay, so this kind of coiling is called as helical fashion, and it is also a right-handed fashion. Okay, the coiling is right-handed, and because of this right-handed helical coiling, you can find the presence of major grooves and minor grooves. And then the pitch of the helix. Okay, this is the pitch of the helix. The pitch means the turn. A single turn is called as the pitch. The pitch of the helix is around 3.4 nanometer, which consists of 10 base pairs, or it is also 34 angstrom units. And then the planes of adjacent base pairs are stacked over one another. Okay, so I think now you understood about the double helical structure of DNA. So hope you had a proper good understanding about the DNA structure, the composition of DNA, the occurrence of DNA in prokaryotic and eukaryotic cell, what are the different bonds or the linkages that are present in DNA, and what are the salient features of the double helical structure of DNA which was proposed by Watson and Crick based on the investigations which were also supportive for them, that is the Chargaff rules and the X-ray studies. We shall further continue the series of lectures for NEET examination. If you have any doubts or if you want to connect with me, you can definitely comment on the comment section. Until then, take care, bye. I will soon meet you in another video. Thank you.